All right, again, welcome everyone to the OpenSim webinar. My name is Jennifer Hicks. I'm the OpenSim R&D Manager and the Associate Director for the National Center for Simulation and Rehabilitation Research. I'll be serving as the moderator for today's webinar. I'm pleased to welcome today's presenters, Dominic Farris, who's joining us from the University of Queensland in Australia, and Greg Sawicki, who's tuning in from North Carolina State University as well. Next slide, please. So a quick reminder about what the OpenSim project is and the goals of our webinar series. The OpenSim is freely available open source software that allows you to visualize musculoskeletal structures and simulate the movements of humans and animals and other structures, as we'll see today. The application includes tools for general purpose inverse dynamics, optimization so you can estimate muscle and joint forces, methods to create simulation for motion capture, and then tools that allow you to analyze and visualize the results of your simulations. So the first goal of our webinar series is to showcase the cutting-edge research that's being performed with the OpenSim software platform. OpenSim is also a growing and geographically diverse community of users. For example, today our speakers are joining us from opposite sides of the world. So the second goal of our webinar series is to provide an easy platform for members of the OpenSim community to communicate and collaborate with each other. Next slide. So before we get started, just a couple quick reminders about the format of the webinar. We will take questions at the end of the webinar, and these will be text-based via the Q&A panel. So go ahead and write down your questions and you'll be able to type those in at the end. If you need any additional technical help during the webinar, you can consult the guide on our website, or you can send a chat to the host of the webinar as well. So with that, next slide, please. Now I'd like to introduce our speakers for today. So first, Dominic Farris. He received his PhD in biomechanics from the University of Bath in 2010. Then he was a postdoctoral fellow at the University of North Carolina, Chapel Hill, and NC State University, working with Greg Sawicki. And then in January 2013, Dominic joined the School of Human Movement Studies at the University of Queensland, where he is a research fellow specializing in muscle mechanics. Our second speaker today is Dr. Greg Sawicki. He's an assistant professor in the Joint Department of Biomedical Engineering at North Carolina State University and UNC Chapel Hill. He completed his PhD in human neuromechanics at the University of Michigan, and he was also an NIH-funded postdoctoral fellow in integrative biology at Brown University. Dr. Sawicki's research is focused on rehabilitation engineering. He directs the Human Physiology of Wearable Robotics, or POWER, laboratory that focuses on uncovering fundamental principles of locomotion mechanics, energetics, and neural control in both healthy and impaired populations, for example, stroke and spinal cord injury. So Greg and Dom are both active members of the OpenSim community, so I'm excited to have them to be a part of our webinar series today. They've both attended workshops in the past here at Stanford, and Dom was also on the Stanford campus for the summer a couple years ago as an NCSRR visiting scholar. They've put together a great webinar for us today that demonstrates several different modeling approaches from simple to more complex, and then they apply these modeling tools to understand the interaction between exoskeletons and the human, which is a valuable and exciting new research direction for our field. So with that, I will let you take it away, Greg and Dom.
Okay, thanks Jen for the introduction, and Dom's going to be controlling the slide, so if you can get us on to slide 1, Dom, that'd be good. Okay, so just a quick intro slide about exoskeletons in general. You can see a number of pictures here of whole body exoskeletons designed to unload muscles during locomotion in an attempt to reduce the energetic demands of tasks like walking, running, carrying loads, going up and down hill for people who are otherwise healthy, and then also to restore function to people who might have mobility impairments. And one limiting factor of these is that despite the large or the long-term goal of reducing metabolic cost of human walking, human movement in general, there's really no published report that a portable device such as these can cause appreciable reductions in energy consumption during functional tasks. And so a major goal of my laboratory is to try to bring to bear a number of tools, experimental, modeling, both combined, in order to figure out how to improve these designs in order to achieve performance goals like reducing metabolic cost. And we think that the main issue is actually bulk of the device, and the fact that they don't really take, the ones that we're seeing here, don't really take much inspiration from how the biological system works. Next slide. So our approach in the lab is one that does attempt to take inspiration from the biological systems that the devices are designed to assist. So you can see here on the top left a cutaway picture of a human lower limb. And what I want you to notice is that it's not just muscles, i.e. motors. You've got a lot of connective tissues in there. And in particular, at the ankle joint and distal joints, you've got long, stretchy tendons that are good for storing and returning energy. And in fact, simple physical models that involve just springs and masses, which you see on the upper right, can explain the center of mass dynamics and actually produce ground reaction forces that are measured experimentally for both walking and running. And so it's our idea, and the idea of many others, which is starting to tack on a bit, that maybe it's a good idea to include springy elements in wearable devices in an attempt to make them lighter, more portable, and potentially more functional. Next slide. And so here's one really great example of this approach that from the literature. This is on the left. You can see a passive elastic whole leg exoskeleton that was developed by Elena Grabowski and Hugh Herr up at MIT in the around 2009. And the idea here is to actually reproduce the springy nature of gait in parallel with the person in an attempt to offload their muscles and hopefully reduce their metabolic cost. There's a little graph, a main graph from their publication in the journal Appl Physiology here that shows the net metabolic power on the y-axis as a function of hopping frequency. They asked people to just hop in place to a metronome at different frequencies. And there are three curves represented here. One, which is labeled in normal and is in diamonds, for normal hopping in the exoskeleton without a spring. And you can see that the metabolic cost is quite high at low frequencies and decreases with increasing frequency. And then there are two elastic exoskeleton conditions that they studied called MLE and SLE. The MLE is a multi-lead spring exoskeleton, so it's stiffer than the SLE, which is a single spring exoskeleton in the open circles. And what I want you to notice is there are a couple really cool things that they found. One is you can reduce metabolic cost by quite a lot, up to 30%, by putting a single lead exoskeletal spring in parallel with a person when they hop. What I think is even more interesting is that the stiffer spring actually doesn't perform as well. And so there seems to be some optimal design criterion behind these sorts of devices that we don't really have a good handle on yet. Next slide. So the question that we're starting to try to ask in my lab is this the optimal solution? And can we do more with less? And two kind of key questions are, could we put an exoskeleton not across the whole leg, but maybe just on one of the three joints? And if we can, how stiff should it be? How does the stiffness influence the performance? Next slide. So really, coming back around to this original framework of study, what I think is missing, and where OpenSim and other simple modeling techniques, and other more simple modeling techniques could come in, is that we need better tools for parameter prescription and optimal functional device prescription. And these tools, not just experimental tools, but also theoretical and computational tools that make the design process quicker and more efficient. Next slide. And so we've sort of embarked on this question, focusing again just on very simple movement, which is vertical hopping. And we started out by trying to understand the underlying physiology of the task by measuring inverse dynamics at each of the 3 joints while people hopped vertically at different frequencies. And what you can see here are these pie charts that we can develop that give us some indication of where the mechanical power is being generated in the positive work phase of the task across the lower limb. So for example, on the left, you see a pie chart at 2.2 hertz. It has 3 pieces of pie, ankle in white, knee in the horizontal lines, hip in the solid back. And what you can see is if you break down the power production across the lower limb, that the ankle is the dominant power source, with knee coming in a close second at low frequencies. And as you increase frequency, the task really becomes dominated by ankle joint. Next slide. Furthermore, we have we use ultrasound imaging in my laboratory, thanks to Dominic bringing it on board as part of our tool set a few years ago. And what this allows us to do is look inside the ankle joint piece of pie and separate out the workload into recoil from previously stored elastic energy in the Achilles tendon and other elastic structures, and then muscle shortening in the fascicles of the muscles. So we made measurements on the soleus muscle, which is the largest in PCSA at that joint. And what we found is indeed during vertical hopping, this is at 2.5 hertz, up to 3/4 of the energy in that ankle piece of pie is coming from elastic recoil and tendinous structures. Next slide. And so we went back to the drawing table and said, well, hey, you know, if especially at high frequencies, if most of the energy generating in this task is coming from ankle, and a lot of that energy is coming from recoil and tendinous structures, why not build a device focusing on the ankle joint with an exotendon attached to a carbon fiber frame, which you see there on the left. And so we completed something like the Grabowski experiment, where we had subjects hop to a metronome with a parallel spring. We didn't think too much about sizing it in this initial study. We picked something that was comfortable for people to hop in, and we performed the experiment. Next slide. And we could generate data that looked very similar to the curves generated by Grabowski and her with a full leg exoskeleton, except we observed something like a dose-response effect. That is, by assisting only at one joint, despite its contribution, its large contribution to the task demand, we only saw reductions in metabolic costs of around 13%. Still pretty good, actually, if you think about it. We're not injecting any energy from active motors or anything like that. The device is streamlined and so forth. You can see below that on the top row are the pie charts for the spring condition. Focus on the 2.5 hertz condition where the minus 13% came. You can see that a majority of the energy in the top row under S is coming from an exoskeleton piece of pie now. So the strategy is that we can supply some of the, meet some of the energetic demand, mechanical energetic demand of the task using the parallel spring, and have good effects even just by influencing a single joint. Next. So with this experimental data in hand, we set out to try to explore the larger design space, and this is an effort by myself and Dominic and a PhD student in our lab named Ben Robertson, who's pictured there on the left. And we wanted to use this simple bouncing task as a way to sort of get at what's going on in the underlying muscle tendon units when you hop with a parallel spring. We could use ultrasound imaging actually to look underneath the skin during spring-assisted hopping. Next slide. And then in parallel, what Ben has been working on is developing simple models. In this case, you can see on the left is a model of vertical hopping that includes only ankle musculature, so it includes no muscles above the knee. And we idealized the triceps surae group as a single lumped uniarticular muscle. So we didn't take into account things like biarticulations. And then we lumped all the muscles together into one muscle that had the same force capacity as all three triceps surae together. And we modeled it as a kind of a standard hill-type muscle model, similar to what you find in the OpenSim platform. And we asked it to oscillate a mass in gravity, which is a simplified way of describing vertical hopping. And what we could do is explore a design space that involved trading off EMG or muscle activation of the human wearer and stiffness of the parallel spring being worn by them as an exotendon. Next slide. And what we found was that even though this model is quite simple, it could capture sort of the basic mechanics of vertical hopping, just as we observe in the laboratory setting. So if you look at force here and you think about comparing the force in that muscle tendon unit to ground reaction forces that you might measure in the lab, you can see a nice smooth rise and fall of force, which is what you'd expect from a spring-like hill model. And you can clearly see a ground contact in an aerial phase where the forces go to zero in the model. And also at the level of the muscle tendon unit, I want you to focus specifically on the power curve from the bottom. You can see that the series elastic tissues in blue are cycling, they're storing and then returning elastic energy. And the muscle tendon unit as a whole tracks the series elastic element quite well, which is what we observe in vivo in humans. Fascicles are operating nearly isometrically during this task. So we had confidence that this model, without a spring in parallel, could capture the essential aspects of the task and let us explore what happens when in the presence of springs. Next slide. And so I'm going to spend a little bit of time here just orienting you to these contour graphs that we like to generate using this simple model. Starting on the left, you see a contour graph labeled injury risk. On the y-axis, at the very bottom, you can see a burst of EMG activity that is quite small. And moving up the y-axis along the left edge, that's different runs of the model where we increase the stimulation amplitude in the Hill model for the ankle extensors moving gravitational load. Across the bottom axis, we're increasing from left to right is stiffness of an exoskeleton worn in parallel with the Hill model. So the left edge on that graph at 0 exoskeleton stiffness going up the line from 10% to 100% activation represents hopping without an exoskeleton at all. And then you can imagine a grid underlying this contour where there are different combinations of biological contribution and exoskeleton stiffness generating the hopping motion. And what we could do then is kind of look at this large space without having to make measurements of many different exoskeletons on many different people and get an idea of what the performance landscape might look like. For example, here on this contour on the left, the hot areas represent places where we saw the biological muscle fascicle get stretched to lengths that could potentially cause injury. They were getting to strains at or above 1.3 L0. And on the right, we could actually find out which combinations of human and exoskeleton generated a flight phase at all. So you can see at the bottom right of the contour, very stiff exoskeletons and not much contribution from the human user generate something more like bouncing and not hopping at the flight phase. Next slide. So the utility of this model continues a bit down to the muscle level. So we can actually evaluate what's happening to the fascicle in terms of its length tension behavior or its force length behavior. And you can see that on the right that we're generating work loops of the contractile element, which can later be compared with ultrasound imaging, which Dom will get into. And all I want to point out here is that if you look at the contour graph one more time, you can see that the power produced by the muscle fascicles, which is what's plotted on the left, it actually doesn't change very much when you trade off exoskeleton stiffness and human activation. So if you follow the track of dots from top left toward bottom right, you can see that there are changes in positive power, but not large changes. And this is kind of an interesting effect, where by putting a spring in parallel with the muscle, we're not actually reducing the work demand on it because its force is going down, but its displacement is actually going up. Next slide. And finally, the real sort of performance variable that we care most about, average metabolic rate here, and also metabolic efficiency, are things that we can sort of gain some insight over using a model where we can model energetic costs, something that you can't measure at the muscle level in humans. And what you can see here is it's actually not that surprising, but the average metabolic rate seems to depend mainly on how much the human turns on their muscles and off activation quite well by increasing the stiffness of the parallel spring in order to reduce metabolic cost. What I want you to notice is these two stars in the metabolic rate graph. Those represent the conditions that we measured in the laboratory, at least as best as we can estimate where those lie in this space. And the real interesting thing here is that in the condition where we have a spring going, right there at about 20% of KSED and 30% stimulation, we're not at a place where it appears in this larger design space is optimal in terms of stiffness. What this model tells us is that maybe we would pick something more like 40% KSED and be able to drive activation down even further and get more metabolic gains. But that after that point, if we increase the stiffness of the exoskeleton, people would not be able to move without a flight phase without increasing their EMG activity again and actually suffering some metabolic insults. So again, this is a very simple model. All the muscles are lumped together, but we think it's useful for giving us something like a coarse idea of what the design space might look like. Next slide. So just to sum up here before Dom takes off, I guess what I'm saying is simple models are a good way to sort of orient ourselves in the larger design space where it might be difficult to make laboratory measurements at every one of those grid points. However, there are a number of simplifications in this model that keep us from getting a real good understanding of what's happening underneath the skin. We don't know what's happening with individual muscles. Our model of energetic cost is for the lump triceps surae, so we don't know whether or not we're affecting, say, soleus more than gastrocnemius. And other things like this that we feel like a more complex model could much better address. And so that's where Dominic's project with OpenSim sort of took off. Next slide.
Giving a great overview of the general topic area. And as Greg said, I'm now going to talk about a specific project that we undertook using OpenSim to simulate the effects of elastic ankle exoskeletons that we've already discussed on the underlying muscle tendon dynamics. And I'm going to revisit a little bit of the background just to clear things up and start by saying that if we look at hopping in place or in typically any bouncing gait, we see really efficient mechanics from the human plantarflexors. And what facilitates that is the elastic Achilles tendon. And the role that plays, that the tendon plays, is that it stores and returns energy, which not only reduces the work that must be done by the contractile part of the muscle, but it actually allows it to operate at low velocities and optimal lengths. So what I'm talking about there, for those less familiar with muscle mechanics, if we look at the force-velocity relationship of muscle here quickly, and we see the yellow curve, which is our force value, we can see that it's easier to produce forces at low velocities. If we then look at also the blue curve, which represents the efficiency of the muscle, it's clearly more efficient to produce forces at lower contractile element velocities. So I guess the message here is that it's good to be at lower velocities if we can manage that for the contractile element. If we then look at the right hand of the two graphs and see the length tension relationship of skeletal muscle, what we can see is that there is an optimal length, L0, represented by the dotted white line, where again, it's easier for us to produce force here. We have to activate the muscle less to produce a given force at this length. And so ideally, that's where we want to be. We want to operate near L0 and at low velocities to reduce the metabolic demands of the task. And as Greg said, people have taken inspiration from these elastic mechanisms to design exoskeletons. And we've seen some beneficial effects of those exoskeletons in terms of things like reduced plantarflexor activation with ankle exoskeletons, reduced contributions of biological tissues to stiffness of the limb, reduced metabolic power, which is our key goal, and reduced joint power contributions from muscles. But the difficult thing is here, we already had a system that was operating really well, and now we've started to change some of the factors like activation, force production by the muscles. So we wanted to ask, how does that feed back into that original muscle-tendon interaction? How do these ankle exoskeletons in particular affect plantarflexor muscle-tendon interaction? And as Greg's already talked a little bit about, we started with an experimental setup, which I'm just going to talk about briefly again, because this was important for our modeling work. And we had 7 male subjects, and they hopped bilaterally for 4 minutes, during which time we were recording oxygen consumption and carbon dioxide production to get metabolic power. We had motion capture and ground reaction forces to perform inverse dynamic analysis to get joint contributions. And we also looked at the muscle level using ultrasound imaging, which Greg showed you, and some electromyography. And I really just wanted to point out that the key messages here of the 2 papers we published from this data were that we can get a whole body metabolic energy consumption reduction with exoskeletons. And this seems to be linked to the fact that we reduce the mechanical demands at the joints. But if we look at the muscular level, although we're reducing the force in the soleus muscle, and we could only look at soleus here, we actually increased the length changes and velocities of the soleus fascicles when we used exoskeletons. And we were unable to alter the amount of work that the contractile element of soleus did. And this was great and interesting, but you'll notice a big gap here between looking at whole body net metabolic energy expenditure versus one individual muscle's mechanics. And it's very difficult to draw direct links between the two. So that was something that we really wanted to strive for, to look at more muscles and to link their mechanics directly to their metabolic cost. And so that's where OpenSim came in as a tool for us to do this. And we set out with several aims, which were, first of all, to develop a model and a modeling framework in OpenSim that we could explore these questions with. Obviously, then we had to implement that framework in simulations in OpenSim. And we then wanted to add to that a model of muscle energetics so that we could predict energy consumption for individual muscles based on the muscle mechanics and dynamics predicted by simulations. We expected that we would reduce forces and activations in the plantarflexors by using an elastic ankle exoskeleton. But we would also see an effect of the exoskeletons of increasing length changes and velocities of the contractile elements of those muscles. And that this sort of negative effect on muscle mechanics would actually limit the reductions in metabolic cost that we were able to achieve in those muscles. So the first step was to obviously come up with our generic model that we were going to use. And in this case, we edited Edith Arnold's model, which is, in this case, a full body model that's available from the simpk.org website. And because we were only interested in the right leg, and in particular the ankle, we removed a lot of elements from this model. We took out the left leg, pelvis, the head, arms, and trunk, and all the muscles except for the 2 heads of gastrocnemius, soleus, and tibialis anterior. These were the muscles that we had electromyographic data for. We updated the muscle models to be the OpenSim 3.0 implementation of the Phelan muscle. And we made a couple of other changes as well to be in line with some experimental studies. So we altered tendon compliance in each of these muscles such that it would match values shown in experimental studies with ultrasound imaging. And we also adjusted the activation and deactivation time constants in the activation dynamics model. There's a table with those values at the bottom, which I'm not going to run through, but what I thought I would do is show you how we changed those properties in OpenSim for those less familiar with the software. The approach that we took, which was actually before the release of OpenSim 3.0, was to edit the model file in Notepad++ as an XML editor. And what you can see here is within the model there is the force set, which contains objects for each of the muscles. Here I've opened up the properties of the soleus muscle, and you can see you can simply come in here and adjust the various different properties. For example, we might want to change the slack length of the tendon. We can just come in here and type that in there. Now, perhaps a better approach to this since the release of OpenSim 3.0 is actually to edit the model in the user interface. And you can see here that if we select our muscle up in the navigator window here, we can open the properties window and we can make adjustments to those properties in the properties window as well. So that was the generic model that we had in place. We then needed to come up with a framework for our simulations, which I'm going to show you an overview of now. So we relied heavily on experimental data in these simulations, and we had processed EMG, joint angles from an inverse kinematic solution, and during our experiments we've measured the torque produced by the spring in the exoskeleton. So those were all inputs to our simulations, and the joint angles and exoskeleton torques were fed into our scaled musculoskeletal model. So we took that generic model and scaled it to each of our participants' data and ran these simulations on scaled models using data for each individual subject. We took the processed EMG, fed that to our model of activation dynamics and contraction dynamics, which form our muscle model. And also into the model of contraction dynamics goes the scaled musculoskeletal model. From there, we're able to then obtain our muscle dynamics that we're really interested in, particularly the force, length, and velocity of the contractile parts of the muscle. From there, we can also take out our activation, take those muscle dynamics, and feed them to our energetics model. In this case, we used a model developed by Glenn Lajoie here at the University of Queensland. And then we could predict metabolic energy consumption and start to make some links between metabolic energy and the muscle tendon dynamics. So that's a general overview of it, but I wanted to also show you how we implemented that in OpenSim. So if we look at our perhaps typical OpenSim workflow, and I should note that we used MATLAB scripting as a tool to implement our OpenSim workflow. What you see here is our typical OpenSim workflow, if there is such a thing, where we would scale our model, run inverse kinematics, inverse dynamics, and then maybe do some residual reduction, run a CMC and the forward tool. Well, we diverted from this typical workflow slightly, and we in fact scaled our model, and then after running inverse kinematics, we took those kinematics and using a MATLAB function that is now available, that is released with the current release of OpenSim, prescribed motion and model, we wrote those inverse kinematics, or the solution from the inverse kinematics, into our scaled model. If you want to see what that looks like or where that goes in the model, again, I've got the XML editor open here, and I've opened up one of the joints in the model, and you can see it's the ankle joint here. And if we scroll down to where there is the coordinate for the ankle joint, within that we have now written a prescribed function in the form of a sim spline, which tells us how that coordinate changes over the course of time during each trial. So having prescribed the kinematics, you might be thinking, well, why did we do that? In the first instance, we only had a limited number of muscles in the model, so we had no actuators driving hip motion, a limited number contributing to knee motion. And so we needed something to maintain the appropriate coordinates for the hip and for the knee. And then at the ankle, perhaps ideally it would have been nice to not prescribe the coordinates and just let the motion be driven by the muscles themselves only. One of the problems with that is when you're dealing with the ankle joint is that the mass and the inertial properties of the foot are very small, and any slight imbalance in the moments between plantarflexors and dorsiflexors, and you'll get something very non-physiological happening like this, which looks rather painful. And it's such a fine balance with that very low mass of the foot that we felt it necessary to also prescribe ankle kinematics. So if we return to our workflow, we can see that we also added the experimental exoskeleton torque values into the model, and these allowed us to model the exoskeleton effects simply as equal and opposite torques on the tibia and the talus. And again, to show you where that goes in the model, if we look at the XML editor again, within our model, we come down to our force set again, and now within the objects as well as muscles, we have prescribed forces. And in this case, you can see again within that prescribed force, we have a sim spline that describes how that torque evolved over the course of time during the trial. We also took our experimental EMG and we did some processing on that. So we bandpass filtered it, smoothed it with a moving RMS, and normalized that data to the maximum value for each muscle, and fed that to a controls file, which was then an input to the forward tool in OpenSim. Also, as inputs to the forward tool, we gave it the scaled model, and we also gave it an external loads file containing the ground reaction forces. So this meant we could skip over these two steps here, and we go straight to the forward tool, which gives us our muscle tendon dynamics, which were fed into our energetics model. We also ran inverse dynamics. This was to help us evaluate our model. So having prescribed kinematics, in order to evaluate our model, we needed to compare joint moments and powers from the inverse dynamic solution to those predicted by simulations of muscle tendon dynamics. And that's what I'm going to show you next. This is how we evaluated our model. So what you can see here are 4 graphs. The 2 left-hand graphs show data for the no exoskeleton condition, where the subjects were hopping without an exoskeleton, or there was no exoskeleton torque prescribed in the simulation. And on the right, it's for the condition where there was a spring-loaded exoskeleton being used. On the top 2 panels are joint moments for the ankle, and the bottom 2 joint powers. The blue lines represent the results of the muscle tendon dynamics simulation, and the yellow lines the results of the inverse dynamic solution. And what you can see here pretty much across the board for moments and powers was very good temporal agreement between inverse dynamic solutions and simulation results, with generally slight underestimates by the simulation of moments and powers. And I suspect this is to be expected, given that we actually didn't scale the maximum isometric forces in the muscle model to individual subjects. And these were young athletic subjects who probably had larger maximum isometric force capabilities than the cadavers that we used to come up with the properties for the muscle model. And so we were pretty happy in general with this. We can see that typically the moments are within one standard deviation of one another, as are the powers. So this gave us good confidence that our model was doing what we hoped it would and representing the experimental data. One of the unique things about our study was that we had another source of model evaluation, which was the experimental classical length data from ultrasound imaging. So what you can see here in both of these graphs is for the no exoskeleton and the exoskeleton condition, plots of soleus phasical length as measured by ultrasound imaging, normalized to hop time, plotted for all 7 subjects. This is the mean of all 7 subjects. And the white line represents the soleus fiber length predicted from the muscle tendon dynamics simulations. And what you can see here in both conditions, we see similar patterns of first lengthening and then shortening during ground contact, which ended at the yellow dotted line there, and similar magnitudes of lengthening and shortening in both conditions as well. So this really gave us further confidence in our model, particularly when we look at the fact that we see similar trends occurring in the experiment and the simulation between no exoskeleton and exoskeleton conditions. So particularly here, I've plotted the amount of shortening occurring in the muscle fascicle during that latter part of ground contact. And in red is the condition where no exoskeleton was being used, and in blue, the spring-loaded exoskeleton was being used. For the ultrasound data and for the simulation results, and that you can see in both cases when an exoskeleton was worn, there was a significant increase in the amount of shortening that had to be done by the contractile element of the muscle. And this is really important when we start to think about what's driving metabolic cost, which is what I'm going to talk about now. We're going to start to look at some of the results from the simulations only now, where we see, again, the no exoskeleton condition in red and the condition where an exoskeleton was worn in blue. And what I'm going to plot here is soleus, lateral gastrocnemius, and medial gastrocnemius data. First of all, for the average force produced by each of those muscles during ground contact of hops. And what you can see here is, as we predicted, significant reductions in the amount of force that these muscles had to produce, which is great. That's one of the things we're looking to do with the exoskeletons, is reduce the loading on the muscles. However, on the other hand, what we saw, which is also as predicted, that the average velocity of each of these muscles during ground contact tended to increase. And particularly for the soleus down the bottom here, you can see that this was actually almost a threefold increase in average velocity during ground contact for that muscle. There was no significant change for lateral gastro, and there was a reasonable increase for the medial gastrocnemius in average velocities. So having looked at velocity, the next thing we wanted to look at was the average operating length of each of these muscles during ground contact. So what I've plotted here is a typical force length curve for each of those muscles, and identified on that curve where the muscle was operating, or where the contractile part of the muscle was operating during each condition. So the red dots represent the average operating length when no exoskeleton was worn, and the blue dots the average operating length when there was an exoskeleton being worn. And what you can see here is, for medial gastrocnemius, a significant and quite large shift down the ascending limb of the force-length relationship when an exoskeleton was worn. Again, no significant changes in lateral gastrocnemius, but a slight leftward shift. And for soleus, a shift, again, leftward down the ascending limb, or certainly down off the plateau of the force-length relationship. So again, largely less favorable things happening when the exoskeletons are worn in terms of average operating length. Then if we look at the activation of each muscle, we can see for medial gastrocnemius, no significant changes at all in activation. This is probably some trade-off between the fact that forces were reduced with an exoskeleton, but then velocities and lengths were less favorable for force production, therefore requiring activation to stay at the same level. Similar finding for lateral gastrocnemius. And if we look at the soleus data, we did affect, or the exoskeletons did affect, a small reduction in activation. But I'd like to point out that this is a relatively small reduction in activation, given how large a reduction in force we observed for this muscle. And this, again, is probably due to large increases in velocity of the contractile element and less favorable operating lengths. The effects of that on metabolic cost, or metabolic rate, we can see we actually did still get a significant reduction for medial gastrocnemius, despite the the increases in velocity and less favorable lengths. For lateral gastric medius, unsurprisingly, no change in metabolic cost, given there was very little change in most other variables. And for soleus, interestingly, we see that there was also no significant reduction in metabolic rate. Again, this seems to be due to this trade-off between the factors that affect metabolic rate, which are the average force, velocity, length, and activation of the contractile element, where we see that force was reduced, but velocity was increased, and operating length fell to a less favorable spot. So interestingly, although we were able to really reduce the forces these muscles were producing, we didn't see any significant reduction in the overall metabolic rate of all these muscles plus tibialis anterior combined. So some specific conclusions from this study would be that, yes, as predicted, we were able to reduce plantarflexor forces with an exoskeleton, but because of changes in fiber velocities, we actually didn't see any significant reduction in overall metabolic energy consumption. You might find this slightly at odds with our experimental result for whole body net metabolic cost, which, if you recall, we did show a reduction in whole body net metabolic cost. One of the things I haven't alluded to here, but I should now point out to clarify that, is that we did see changes at other joints when the exoskeletons were worn as well, particularly the knee. There was actually a reduction in work done at the knee joint when exoskeletons were worn. And so this plays into that total whole body net metabolic cost as well. Some more general conclusions, trying to draw back to what we were talking about at the start. We've seen here that we can use complex musculoskeletal models, particularly in OpenSim, to look at specific mechanics and energetics of individual muscles. And what this has highlighted is that designers of assistive robotic devices really do need to consider muscle tendon level dynamics, not just joint level mechanics, when they are assessing how well their devices are performing. We may affect changes at joints other than the one we're assisting when we use assistive robotic devices. That should also be considered. And these models can provide an excellent framework for exploring other design parameters. So in the same way that in Greg and Ben's simple model, they were able to vary things like spring stiffness, we could vary that in this OpenSim model by adjusting that torque that we applied to represent the exoskeleton. I'd like to finish by acknowledging some people, particularly the National Center for Simulation and Rehabilitation Research. As Jen said earlier, I was lucky enough to be a visiting scholar with them, which greatly aided this project, and all of the OpenSim team who have been extremely helpful in advancing this project. I'd also like to thank two of the PhD students at UNC Chapel Hill and NC State, Bruce Wiggett and Ben Robertson, without whom these projects would not have been a success. Thank you.
A great talk. We'll now dive right into the Q&A session. So as I mentioned earlier, all of the questions will be text-based. So you want to find the Q&A box next to the Q&A panel in your WebEx controls. Type in your question and make sure you select to ask all panelists so that we can see your questions come in. Okay, oops, let me bring mine up too so I can see the questions. Okay, so while we're waiting for the questions to come into the from the audience, I was wondering, so you mentioned that the knee was also affected by wearing the exoskeleton, and that the work done at the knee went down as well. Have you looked at if you had EMG from any of those, or do you those muscles, or do you have any speculation about what muscles might be affected at the knee?
We don't have any data for the muscles at the knee. If we'd had EMG data for muscles crossing the knee, it would have been nice to have included them in the simulations. Like I said, all we know is that the joint work at the knee actually went down. So there was a bit of a shift in terms of loading from the knee to the ankle, where presumably it could be borne by the exoskeleton. So in terms of, it's difficult to say what might be going on at the muscular level, but it would certainly be interesting to see if we were actually getting, by that reduction in work, whether we actually got a reduction in metabolic cost from muscles acting at the knee. As we've seen, reducing the work doesn't necessarily reduce the metabolic cost, so we'd have to look at, again, this is where models like OpenSim models can come in and help us actually look at the contractile element mechanics.
Well, I guess even taking a step back, I think it might be counterintuitive for a lot of people that a device at the ankle would affect what's going on at the knee. Can you explain a little bit more just about how that can happen?
Sure. So, I mean, what I suspect is happening is, like I say, people hopped for a period of time in these exoskeletons, getting familiar with them. What I would suspect is, especially having hopped in them myself, is that once you feel that you can allow the exoskeletons to take over some of that loading, first of all, you're going to allow it to take over some of the loading at the ankle. But if you can then adjust to find a strategy where you could shift work from elsewhere in the limb to that exoskeleton, then I think you would preferentially choose to do that as well. So you might start, like say, using your knee less. The hip really isn't doing very much, or there's not much happening at the hip here in terms of work. But certainly if you could find a way of shifting work from the knee to the exoskeleton by changing the kinematics of the movement, which is actually what we saw, then yes, perhaps we adapt our control strategy so we can make the most of the device that's only helping at one joint.
Okay, thanks. Now there's a question from Taylor Dick. To what extent do you think that the performance of these exoskeletons or accuracy of the models is dependent on subject-specific tendon properties such as slack length and stiffness? And did you explore this at all?
And one of the reasons why, as you saw at the start, we went in and adjusted tendon properties was because those properties do make a big difference. If you don't have the correct, well, I'm not going to say correct, if you don't have an appropriate compliance set for those tendons, then you are going to see quite different muscle mechanical behavior. We haven't done ourselves a sensitivity analysis on this. I believe, Jen, you might be able to support me on this. I think, did Edith Arnold, I think, do some sort of sensitivity analysis on the effects of tendon compliance?
She did look at the effects of tendon compliance on fiber length and velocity during walking and running. And there are some muscles that are more sensitive than others, is the short answer. I think some of the plantarflexors are sensitive to the properties that you choose.
Yeah, and so yeah, it's important to try and get appropriate compliances. As for doing it on an individual basis, again, I think this is where you get into the realm of the utility. I mean, it would be nice to, for every single property of the muscle, determine an experimental value for each person. But if we're starting to do that, then we're probably starting to get away from one of the main utilities of these models, which is actually that you don't have to collect large amounts of experimental data on every single person. You can actually start to run simulations with without having to do huge amounts of experimental data collection, so we can explore design space and so on. That would be my answer to that.
Okay, yeah, thanks. Let's see. So audience members, go ahead and type in more questions for Dom and Greg. While we're waiting for those, I'll ask another question. So do you have, I guess, how can you, maybe you already said this, but how did you choose the stiffness of the device that you tested? And how much of an effect do you think that if you vary the stiffness, how much of an effect would that have on the muscle tendon dynamics during the hopping motion?
As Greg hinted at, I think in the experiments at least, the stiffness that we chose initially anyway here was one that could be used comfortably. So if you put a very stiff spring in, people can't hop at all in it. So we basically went for the stiffest one, the stiffest spring that could be comfortably hopped in, under the assumption that that spring would be giving the most assistance. As for the effects of it, perhaps I might defer to Greg here to talk a little bit about some of the more simple muscle simulations, which explored that.
Yeah, so Dom, if you can page back to the slide with the metabolic cost contour, I think we don't know for sure what would happen in the ankle device. What we do know is that when Elena and Hugh Herr put a stiffer spring on their whole leg device, that the metabolic cost actually did not drop as much as with a more compliant spring. So it seems like there's some motivation that there might be a sweet spot. You don't want to be too compliant, but you also don't want to be too stiff. But if you look at the contour that Dom's brought back up here from our much more simple lump model of the plantar flexors, you can see that along the bottom where the border of injury risk and no flight exists, what this model would predict is that right around 40% or so of the tendon stiffness, you could avoid injury, hop with a flight phase, and get a slightly better metabolic reduction than what we think we have with the device that we're currently using. But if you made things stiffer than that, people would have to turn their muscles on more to keep hopping. So I don't know exactly where the value is, and our best estimate is that we're at the 2-star conditions in this space. But these are rough estimates. So I think the next studies are to actually go back into the lab and play around with different stiffnesses that, cover the range along this x-axis that we're showing.
Yeah, that would be cool to see. Okay, so we have time for a couple more questions. We have another question from the audience. This is Taylor again. What was the rationale for collecting experimental data on the soleus rather than the medial or lateral gastrocnemius?
Major motivation for looking at soleus was actually that it is the largest by far in terms of physiological cross-sectional area of those 3 muscles. So in terms of trying to link what's happening at muscle tendon level to the joint level, and then potentially even, although you're getting into slightly shaky territory here, to the whole body net metabolic level, we felt it was best to choose the largest muscle, which represented the muscle that was producing the majority of ankle torque, doing the most work at the ankle joint. It's also slightly less complicated in terms of the fact that it's uniarticular, so it's less affected by anything that might be changing at the knee, which is good. It is. I should also note it is slightly harder in terms of the ultrasound imaging though to really get good images of soleus. So in some senses, gastrocnemius, or in that sense at least, the gastrocnemius would be an easier option.
Okay. All right. Well, if there are no more questions from the audience, can you guys go ahead and bring up the last couple slides for me? the end of the talk. So as you'll see in the slides, I also want to acknowledge the funding sources for the OpenSim project and the NCSRR. OpenSim and this webinar series are supported by several grants from the NIH and the EU, including an NIH grant that funds our National Center for Simulation and Rehabilitation Research. We're also supported by the DARPA Warrior Web effort. Next slide, I think. Oh, yes, there are our actual funding sources listed there. So I really want to thank everyone for a great discussion. All the great questions coming in and having speakers all over the world is really exciting. You can find information about upcoming webinars, past webinars, and other upcoming events and resources on our website. So that's opensim.stanford.edu. And we also ask that you please complete the survey that will appear in a pop-up window at the conclusion of the webinar. This will help us improve the webinar series and also choose upcoming topics. So thank you again to Dom and Greg for putting in the work to prepare this great webinar, and thank you all for tuning in and participating. We hope that you'll all continue to stay involved with OpenSim. Thank you, guys.